Okay, in case you missed something during the first set of lectures, this is BCH 190 and its attendant cross-listings, issues in biotechnology. I prefer to call it actually the way we work with life. And in the first lecture, I covered an overview about what we will be covering in this course. And it's set up basically in two major sections. In the first section, I intend to cover how life works. That is, the mechanics of life. And how do we work with it? What are some of the techniques that we use in biotechnology? In the second part, we will look at the applications in biotechnology, agricultural biotechnology being first, and then we'll look at pharmaceutical biotechnology, medical biotechnology, and all of its subtopics, including cancer biology, pandemic flu, bioweapons, and modern applications. And we will have a section on forensics and its modern applications and marine biotechnology. So in tonight's lectures, I want to really get at the foundation of how life works. From the mechanics on out. How do you go from atoms out to molecules, out to an information that contains, out to a molecule that contains information like DNA, out to an organism that's capable of speaking about it. In the second part of tonight, I'll cover briefly some of the techniques that we will be introducing in biotechnology. So first, from atoms to cells to the flow of life. In the first part of the lectures last week, we looked at science as a system of hypothesis making and testing, as a way of getting at knowledge. And I said that maybe this is the best way that humans have come up with yet of deriving knowledge. Certainly not the only way, but perhaps one of the better ways. And what we're going to be really encouraged to focus on in this class is evidence-based knowledge. We can take lots of other courses that have to do with beliefs and thoughts and philosophies, but really what we want to try to do in this class is look at evidence-based knowledge as related to life and biotechnology. So to do this, basically we will be relying on peer-reviewed journals. And here I've listed some of the top peer-reviewed journals in science. And when I say peer-reviewed, sometimes this is misunderstood as reviewed by a bunch of your friends. They're hardly your friends. Peer review is really a consortium of people that are capable and competent to review your work. In other words, as a scientist, if I send out my work to be published, it's going to be harshly criticized by my contemporaries. And either it will get accepted or rejected. And basically, to make it into a peer-reviewed journal is certainly an uphill battle, and it's something that is not to be taken lightly. So sometimes I hear in the popular press, oh, that was just a peer-reviewed journal. Oh yeah? You go ahead and try to do it. So here I've listed really the top 4 highly respected scientific journals published in the world. Science covers all topics from particle physics to astronomy to molecular biology and biotechnology and everything in between. I would say it represents our best view of scientific facts. Realize that scientific facts are changing as we accumulate more knowledge. And this ranking is based on their impact factor. Impact factors in science are related to how many people cite your work. If it's no big deal, then who cares and it's not cited. If it's cited often, it will be repeated. And if it can't be repeated, then it also becomes junk. So basically, to make it into one of these journals is difficult, and to remain a fact over a period of time is a testimony to what we would say is factual knowledge. So when we consider all of these controversies in this class, really what we want to do is rely on the best peer-reviewed journals. Sometimes I will be referring to popular press, New York Times, Time Magazine, and so on, because it's good to know what the public thinks about these things. So science then is a system of making hypotheses and developing experiments in order to test them. If not this, then this, and no other options. And by this, we can discern and validate observable facts over time. This seems to remove the creative process from science. I would like to testify that that is certainly not the case. Oftentimes, scientists are viewed as somewhat nerdy, and not maybe the wild creative types like, say, Mozart and John Lennon might be, or Jackson Pollock. But on the other hand, I would say that creativity in science is very much the same. It's just the medium is different. Science allows us to have ideas in which they can be actuated. Oftentimes, artists will suffer their whole life not knowing whether or not their creation actually worked. Some of them, like Van Gogh, might cut an ear off over that problem. Whereas in science, you can say, ooh, I know, and we can do it and test it and see whether or not your idea could be actualized. The medium is somewhat different, but the creativity is nonetheless the same. Okay, now we're going to get into this question a little more seriously. We touched on it the last time. I want to get right down to it in the first part of the lecture tonight. What is life? You can read this out of the Webster's Third Edition Dictionary. Life, pronounced life, is a noun, a condition or quality that distinguishes a vital and functional being from a dead body. There you have it. I want to elaborate on this just a little bit. I want to come up with a more refined definition of life, and you can read all kinds of things. Life is a day at the beach. Life is short. Life is what you make it. I want to have a better understanding about the mechanics of biological life. And I hope in doing this, it'll change the way you look at all living things. Really. I will start off with the idea that life, biological life, involves information. And that biological information is copied and replicated and processed. So I might start off with a loose definition that life is an information processing system capable of replication. So my computer is an information system, and in some ways it's capable of replicating through cut and paste functions, but I want to elaborate on how biology does it, and then look at how that differentiates itself from other forms of information that you're perhaps more familiar with. The information processing system in life uses the molecule DNA, and its information is then transferred to an intermediate molecule called RNA, which is then further processed into another language of proteins. And it's this interaction and this communication through which life itself functions. But how did this all begin? You look around you and you say, my gosh, this is so complicated. This is so complicated. How did all of this start? Did it start here? Is there life on other planets? How long did it take to get like this? and how does it work? So for this week, you should be looking at the second chapter in the Phelan text, What is Life? And this is primarily on chemistry. And I want to give you a brief background into this just to make sure we're all on the same playing field. A lot of this is review to what you've already had, but let's consider this as the basics, because actually life is chemistry. And below that, life is physics. So how did all of this happen? Well, if you take physics, and if you believe in Einstein, since the Big Bang, all of this is set in motion. And we recently, this summer, on July 4th, 2012, saw the Higgs boson for the first time. If you don't know what that means, look that up. This gives serious validation to what happened about 13.6 billion years ago, and particle physics in general, and the general theory. So let's consider the key to elements, the periodic table. I had mentioned the last time that there is one periodic table in the universe. Why is that? Because of physics. There's not another periodic table out there. The periodic table is the periodic table in this universe. How do we know that? Well, it's related to the physics of the particles that surround these atoms. So if you're thinking that there might be life on another planet, it has to involve our periodic table. And there are even elements, you know, on that periodic table that didn't exist before, or at least not here, before humans had something to do with that equation. So when we consider life on this planet, the central atom is going to be carbon. And this is pretty important, because you can't consider another atom which has the multiplicity of chemistries that carbon does. You can't imagine, for example, a life system that is based on lead. You simply don't have the types of molecules that you can make and the versatility that carbon has with sharing its electrons. Carbon can share its electrons with hydrogen, phosphorus, sulfur, oxygen, nitrogen, and other atoms. Silicon is pretty good. You might think, and some people have, about alien life existing on a basis of silicon chemistry. Not too bad. We can almost get there with that idea. So the periodic table of elements is the same throughout the universe, and this predicts that life would be based on carbon chemistry because of the multiplicity of molecules that can be made using that as its basic Lego set. So atoms are elemental units out of which everything is made. We have to start with that because, you know, I don't know about you, but I've never seen an atom. I kind of am going with the idea that they exist, though, because, you know, there's a lot of good evidence out there for atoms, and they tell me even that this podium is made of molecules that contain atoms, and there's a lot of space, and there are electrons that are moving inside of this thing, and this is kind of hard to conceive, but there's a lot of evidence that supports that idea. You can go out and read about that outside this course, or we can start right from there. I suggest we start from there. These diameters are from 1 to a few hundred millionths of an inch, so we're talking very small scale, and the particles that exist are arranged around a central nucleus. So we have electrons that are moving around a central nucleus. The nucleus can be composed of protons and neutrons, and hydrogen is itself an interesting atom, however simple. If you ever take a course called physical chemistry, you will spend one entire semester on the calculus of hydrogen. And that's not an easy class. And it's not an easy atom to even to understand when you get right down to it. It's one electron moving around a proton. So if you can get your head around that, then the rest of the periodic table might fall in order. Carbon, on the other hand, is composed of 6 protons and 6 neutrons in its nucleus and 6 electrons moving around it. the probability of where those electrons actually exist are not in orbitals exactly like, well, we do call them orbitals in chemistry, but it's not exactly like the orbits of a planet around the sun exactly, but there's a probability of where those electrons would be around that central nucleus. And ions are charged atoms, where a donated electron from one atom might go to another atom, and therefore these ions can be attracted to each other in this loose charge association called an ionic charge. So that sodium, for example, might have a net positive charge and chloride a net negative charge. That reminds me. There was this sodium atom that walks into a bar, sees this chloride ion, and starts chatting it up, you know. And all of a sudden the police come in, and they say, "You're under arrest." And the sodium ion says, "Why?" He says, "That's a salt." That's why I'm a biologist and not a comedian. But nerds tell jokes too. So anyway, that's how salts are made. Covalent bonds. Yeah, I won't do that again. She warned me against that. Covalent bonds, on the other hand, are made when electrons are actually shared. And this is a more serious association in the sharing of electrons that make these covalent bonds. So we have ionic bonds that are shared, that are associating ions when they're brought together. And we have covalent bonds that are made when electrons are shared. And there is another association that I want to acknowledge where a molecule like water has association with other molecules of water through the charge of its associated hydrogen atoms. So if you have oxygen as a central atom and one of the hydrogens is associated with it, the electron is going to be more strongly drawn to the oxygen atom, leaving a negative charge sort of hanging out there. The reason I want to bring this up is that water is actually pretty strange stuff when you get right down to it. Even though it's all around you and you think it's quite usual, it's anything but usual. Because of this polar property, it sticks together. At any one moment, water is almost a lattice. It's almost a crystal lattice by the sharing of these hydrogen bonds. The thing is that it shifts so quickly as to remain a liquid. Water has also other interesting properties. It expands when it freezes. Most compounds don't, you know. They contract and they sink. Where would we be if that were the case? Also, water is very cohesive. If you were to take two planes of glass and sandwich water in between, its tensile strength is greater than that of steel. Try to pull that apart. This cohesive nature allows water molecules to stick together so that they can be transported to the top of a sequoia tree. So water sticks together, it's very polar. It also can dissolve molecules. It's, after all, what we call the universal solvent. You can throw salt into it, and it comes apart into sodium and chloride. But also, water dissolves most of biological materials. DNA, RNA, proteins, all dissolve in water. So the chemistry of life dissolves in water. That's why we're looking for water on Mars. Or we consider that water, with a carbon chemistry, is probably required for life. So if you're into science fiction and you see all these weird creatures in these movies, you have to consider that they had to have at least these constraints, and probably a few more. So, like the atoms in this podium, do you believe in things you can't see? I brought up the last time that we're used to thinking about our reality based on our senses. And the human senses, in spite of what you might think, are really quite limited. You know, your visual perception is limited to the visible wavelength of light. you don't see so well in IV, I've noticed. UV or infrared. So on both ends of that spectrum, we are losing a lot of information. And we're losing a lot of other information too. Humans don't smell so well. Look at what a dog can sense versus yourself. And there's lots of other ways in which we do not sense our reality as well as you might think you do. Nonetheless, we are convinced that this is reality. But if you could see with, say, atomic eyeballs, this podium in your reality would be quite different. So let's just put this into a scale that is relative. An atom, if an object is more familiar like a BB, would then make a simple molecule, say like a glucose molecule, like a marble, whereas a chained molecule, a complex carbohydrate or a strand of DNA, might be similar to that size of a cat. A ship might be the size of a cell, a tractor-trailer, a molecular structure. And a mountain range like an organ, and a continent like North America, a multicelled organism. So is water required for life? Basically, based on the properties that I just told you, very likely. If you can think of another solvent that might do, you would have to work out the chemistry around some other atom other than carbon. And we think that carbon chemistry is required for life because of its versatility in making molecules out of sharing electrons with other atoms. So if we stick 2 atoms together or 3 atoms together, we might make water as a simple molecule. But in biology, we can also put simple molecules together, making sugars, nucleotides that are the basis of nucleic acids like DNA, or amino acids that are the components of proteins. So carbohydrates are made of sugar, like glucose, which is a 6-carbon chain sugar. And it is the energy in these covalent bonds between carbon and hydrogen that store a great deal of energy that can be easily broken down by organisms and translated into chemical energy for cells and organisms. So if you were to say, and people have misconceptions all over the place about nutrition. Humans spend a lot of time thinking about what they need to eat or not eat. Especially in this country, it seems to be a fixation. And we'll get into that later when we talk about agriculture and periodically throughout this course. So which of the following foods is not a significant source of complex carbohydrates? And I don't know if I even really like this question. This was out of your textbook, and I'll explain it in a minute why I perhaps don't like this question. But people have misconceptions about the food that we eat, and I think that's illustrated all over the place. What should you eat? What's good for you? What's not good for you? Nutrition, exercise, fitness throughout our life are long-lasting questions in our society today. Really, you know, I like that answer, but in your book it's going to say that fresh fruit is not a complex carbohydrate because it has simple sugars primarily, but it does have cell wall material and other carbohydrates in it. So I just wanted to point that out as sort of a dumb question in your textbook. But structure of proteins. Proteins are made primarily of amino acids that form the primary structure of amino acids stuck together, and these form a secondary structure into shapes, and a tertiary structure which is somewhat more complex, and a quaternary structure when one or more of these proteins are bonded together. And it's the shape of the protein that's important, but it's the linkage of those amino acids which give it its shape. I'll get more into that in just a minute. Lipids are also another important component. These fats comprising sterols and phospholipids, make up the membranes in cells and are very important. Nucleic acids are made of these building blocks of nitrogen-containing bases that I'll allude to more in a minute. Adenine, thymine, guanine, and cytosine. Together, we have a class of molecules in biology which are made up of chains, whether this is nucleic acids like RNA and DNA, or proteins that are made of polypeptides, or complex carbohydrates that are made out of sugars. Together, these can all make molecular structures like proteins, but larger structures such as ribosomes, parts of cells that are involved in making proteins, or organelles like chloroplasts and mitochondria. So inside of a cell, which is the basic unit of life, we have these different compartments. And it's interesting, I think, that life is compartmentalized. Many of us do this in our own life anyway, especially these days. But life itself is compartmentalized. We have different structures in cells where different things go on. And this turns out to be a hallmark of life itself, compartmentalization. If you're not very good at it, you're not going to do well in these days, I can tell you that. But so it is with a cell. All these different structures we call organelles have different biochemical reactions contained within them that are specialized. A centrifuge is, and I'll talk more about this in the second part of tonight, is a laboratory tool that's used for separating out components of the cell based on their size and density. Think of it as a washing machine on spin cycle. The heavy things will go to the bottom of a tube, spun in a centrifuge, as the lighter things will stay at the top. And we can separate out things on that basis. For imagine if you took this cell that I just showed you a minute ago and broke apart its components, the nucleus, the central part, is the largest, and it'll go to the bottom of the tube very quickly. Lighter components, mitochondria, vesicles, and things will stay more towards the middle, and the lighter components will be at the top. I've spent an inordinate amount of my life watching an ultracentrifuge spin up and spin down. That's as nerdy as my joke, I guess. So the structure of DNA. In order to get at really a foundation for understanding life and its flow in information, we have to know about DNA. And we've all heard the word gene. What is a gene? A gene actually is a piece of DNA which is the code for a protein. So genes are segments of DNA specifically that code for proteins. You have about 24,567 genes or so that code for a human being. That's only about 2 to 4% of your DNA. But proteins do all the work in cells. They function structurally, like in tendons and hair and muscle tissue. They function metabolically in chemical reactions to catalyze reactions to break down metabolites. They move things around inside of cells and between cells. Think of neurotransmitters. They're regulators which function to turn on genes and turn off genes. There are defender molecules like antibodies in the immune system which recognize self from non-self and defend us against invaders. And there are communicators like hormones, which communicate signals to distant parts of cells and organs. All of these are proteins. So by controlling the information that controls proteins, we control the functions of cellular life. So that's what a gene is doing. A gene is controlling the functions of a cell and life by regulating when these proteins are activated in certain cell types. So the central characters then are DNA, RNA, and protein. But first, let's take it from a cell on up. I said that a cell is the basic unit of life. All the information for each protein in a cell is in its DNA. Most of the DNA is localized in a cell nucleus in higher organisms. So all the information that codes for a complete organism is in its DNA. We know that now from the cloning experiments that have been done on various animals. So we have to start somewhere. So let's take the Leeuwenhoek approach and say that the cell is the basic unit of life. We can classify cells into two major groups. Prokaryotic cells, kary meaning nucleus, pro being before, so before nucleus. Prokaryotic cells are mostly microbes, so you have bacteria that comprise most of this group. And this is a very important group, probably the first group of cells on this planet, and they will be probably the last group of cells after the lights go out. They've poured themselves into absolutely every single niche on this planet and can survive in one way or another on all kinds of bizarre chemistries. If you want to say there's an alien on Earth, it might look like this. So prokaryotic cells have been around for about 3.6 billion years or so. Humans, a brief second. So these organisms are tried and true, very successful environmentally, as well as their survival through time. In fact, if you take a human, there are 10 times more bacterial cells in a human than there are human cells in a human. And we'll get into more of that later. So in fact, if I'm an alien looking at you, I say, what an interesting vesicle for prokaryotic cells. So these are single-celled organisms. They lack a membrane-bound nucleus. They have one internal compartment, and they're mainly the bacteria, the eubacteria and the archaeobacteria. But let's not be fooled. They're by no means simple. They might be simple just from your point of view, but you're a little bit anthropocentric. They have an outer membrane, they have a cytoplasm, they have ribosomes for making proteins, and they have DNA and RNA, and basically all the stuff of life. They do lack a membrane-bound nucleus, but they have a nucleoid, so-called, which contains the DNA. All of the DNA copies every time one of these cells divides. We look at basic higher plant cells, shown here, characterized by animal cells and plant cells, but let's not leave the fungi out of this. It doesn't fit on this slide, but the one thing that we notice, the difference between these cells, which we call eukaryotic, EU for true, kary, nucleus, true nucleus. So these are characterized by a large central nucleus, which contains most of the DNA, but not all of the DNA. There is some DNA in animal cells within the mitochondria, and in plants, there's some DNA in chloroplasts. So if you look at plant and animal cells, they contain most of the same internal structures with a few exceptions. Animal cells do not contain chloroplasts, and plant cells are also bound by this cellulosic cell wall, whereas animal cells stick together adjoining their membranes. Other than that, a lot of the functions are very similar. So a eukaryotic cell, remember, contains these various membrane-bound compartments, and it's within these compartments that different biochemical reactions occur. In mitochondria, for example, one of its major functions is respiration, breaking down sugar into the chemical energy stored in ATP. So, for example, muscle tissue will contain a lot of mitochondria for that function. Chloroplasts present, oh, I should point out something unique about mitochondria and chloroplasts. They're the only organelles in a eukaryotic cell that in fact have two membranes, an outer membrane and an inner membrane. Other vesicles, primarily like lysosomes, vacuoles, and things like this, are bound by a single membrane. Let's keep that in mind for a minute. Chloroplasts function in photosynthesis, arguably the most important chemical reaction on the planet. and that all biological energy comes to us through that reaction. Animals and other organisms are entirely dependent on the energy that comes to the earth biologically through photosynthesis. And the photosynthetic reactions then are contained within the chloroplast, which also has two membranes. So there you have it, eukaryotes and prokaryotes, and I'll leave that description more up to your book. But where did these organelles come from? This was a question that was posed in the early 60s after people were looking in the electron microscope at eukaryotic cells. And people like George Palade and Albert Claude and some other people who won a Nobel Prize for describing these had posed the question, how do these different organelles form? How does an endoplasmic reticulum or a Golgi body form within inside of a cell? But one of the things they wondered was, where do mitochondria and chloroplasts come from? And it was suggested that the membrane, the plasma membrane, invaginated to form these two double membrane bound structures, commonly called the plasma lemma invagination theory. On the other hand, there was a woman at UMass Amherst, just right over here. Her name was Lynn Margulis, who she said, you know, I don't think that's right. I think rather what happened was that mitochondria and chloroplasts represent an ancient event where one cell ate another prokaryotic cell. And that prokaryotic cell came to live within its host, or actually within its predator. And everybody in the day said, Lynn, you must be crazy. She called this the endosymbiont hypothesis, that this prokaryotic cell had become an endosymbiont, living within the cell that ate it and functioning together with it. So people argued about this for a good 10 years or so. So this describes these two ideas. In this one, Lynn Margulis is saying the endosymbiont hypothesis, where this ancient eukaryotic cell, here with its nucleus, eats this poor prokaryotic cell, which actually survives this, this attack and lives with inside it and even shares some of its DNA with it, and they mutually benefit. In the plasma lemma invagination theory, this membrane enfolds to eventually become these structures. Well, it turns out that Lynn Margulis went on to say that not only did perhaps this happen just with the development of mitochondria and chloroplasts, but perhaps flagella, perhaps lots of other structures in cells came to develop from one cell eating another cell. So, given the advent of molecular biology, Lynn Margulis has been more than vindicated. In fact, we now understand that mitochondria and chloroplasts have their own DNA. And that DNA looks remarkably more like prokaryotic DNA than it does eukaryotic DNA. They have their own ribosomes that happen to be the same size as the ribosomes in bacteria, not in eukaryotes. It uses the same protein-making machinery as bacteria do. So in fact, when you read the list of prokaryotic-like characteristics that mitochondria and chloroplasts have, we think entirely that these represent an ancient endosymbiotic event. So today, most serious biologists concur that chloroplasts and mitochondria probably represent an ancient event like Lynn Margulis described. So the endosymbiont hypothesis pretty much is where we feel now that these organelles had their origins. So what is life? I will suggest that this is not just a one-off or two-off kind of an idea, but actually this happens all over the place. This borrowing, begging and stealing into a confluence of life. You yourself are not just made of human DNA. I can tell that by looking at you. There's some virus, there's some other organisms, in fact, involved. So, in fact, life itself is this collage of information that moves through it. And so no organism itself is unique. Even the genes that you have are mobile and move around, borrowing components from each other. So from information to function, that's what we need to understand. What is life? I would suggest that actually, given Lynn Margulis was right at least once and probably twice, she wrote this book with her son, Dorian Sagan. She was married to Carl Sagan for a while. And they wrote this book, What is Life? And so given that this is one of the central challenges of these first few lectures in this course, I would invite you to read this book because she has some commonalities about life that you might not consider. She would say one of the commonalities of life is consciousness, for example. Let that bend your head, just in case you thought that humans were the only one. Or let it bend your head in terms of what you think consciousness really is anyway in a human. It must have a biological function. It must have a biological foundation. Why are you thinking about the words that I'm speaking now? How are you even hearing the words that I'm speaking? But okay, this begs the question then, what is death? Like I said in the first lecture, you're here 1 minute, gone the next, but all your molecules are there. What happened? And this has bothered people for a very long time. The first ritualistic treatment of people were over death, leaving trinkets, burial rituals, art pieces. You know, Jack was here just a minute ago. All his stuff's still here, but Jack's gone. What was that? So this confluence of life has also intrigued other people. So it's not just the endosymbiont hypothesis, or it's not just you as a human. This blows out into the ecology of the planet. In the 1950s, NASA contracted this guy named James Lovelock to analyze spectrographs of planets in terms of their atmospheric content. And they asked him, they said he was an atmospheric physicist in the UK, and they asked him to come up with a report about whether or not there could possibly be life on other planets based on these atmospheric analyses. He wrote back a report to NASA that basically stated that his conclusion was that the planet Earth was an organism. This is not exactly what NASA wanted to hear, but it had all the properties of an organism. The planet Earth functioned as an organism. So we ourselves might be just a part of a larger organism the way that mitochondria are a part of us. So in this whole ecology of things, nothing happens in a vacuum. So in chaos theory, Stuart Kauffman wrote, you know, the famous line, "The butterfly beats its wings in the Amazon and changes the weather in Chicago." It's all connected, you see. And this is so relevant these days as humans have tweaked this thing beyond control. So Lovelock called it Gaia, which is an old term, actually. This has been around for a long time, paraphrased otherwise as Mother Earth. And he has since written other books, The Revenge of Gaia, Why the Earth is Fighting Back, and more recently, The Vanishing Face of Gaia. Why is DNA referred to as the blueprint of life? Are you just a function of your genes? I couldn't help it. My DNA made me do it. Has actually been a defense in certain courts of law. How is information stored in the molecule of DNA? How is it decoded? And then how are genes cloned now? And what does this mean? We went over the last time the difference between belief, fact, knowledge, truth. These are all simple terms that you think you might know. But like a lot of other words, sometimes words that you think you are very familiar with deserve a look at. Sometimes a dictionary can be very entertaining that way. So belief is a mental act or condition, or even a habit, of placing trust or confidence in a person, a thing, and it usually involves faith. Notice the word knowledge or evidence is not in there, and it may or may not be involved depending on the context of how that word is used. Do you believe in elves? I mean, seriously, little people with pointed ears that do magic. Whoa. Okay. If you have one, bring them in. I'm sure we'd really like DNA evidence would be good too. How about Loch Ness Monster? You familiar with this? This is a beast. There's photographs, fuzzy at best, living in Scotland, maybe. Is there a Loch Ness Monster? 28%. Okay. 47%. What was it? 16 in the elves. What about Bigfoot? This would be, it could be Yeti's abominable snowman, the one that's out in Oregon maybe, or Alaska. Same bunch of people. It actually went down. More people believe in the Loch Ness Monster. Maybe. How about aliens? And this would be from another planet that have actually landed on the Earth. That would be my criteria. Like elves, little people with pointed ears that do magic, I want aliens that actually have landed here. They might be out there, but I want ones that landed here. Yeah, okay. Do you know, this is in line with national average. In fact, this class is doing pretty good on these questions so far. If you read about this in national polls, this is pretty much a good demographic for what we see on these questions nationally, and it's actually representative of what I see in the past in this class, except that I've seen more people believe in elves previously. So, how about ghosts? About the same. One last one, okay? An everlasting human soul in an afterlife? Okay. That went down. Did you see that? More of you believe in ghosts than an everlasting human soul. That's interesting. Okay, the reason for that exercise is I don't think that there is, there's not a peer-reviewed journal that carries evidence for this type of thing. In other words, we don't have DNA evidence for elves. There's been no Loch Ness Monster that's washed up on a beach yet. Some fuzzy photographs. Same thing with Bigfoot, and the rest of it is kind of up to you, really. It's kind of subjective, but it all falls into this category of belief. We might say the same for a higher power, but what we want to look at now, I want to differentiate all of that from the function of life on this planet. What do we know about this stuff, right? not fuzzy photographs, not just some hearsay. What can we actually look at and understand? And concretely, how can we use that information to affect us concretely? So what is DNA? How does it work? What is a gene? What is a clone? Well, you know, typically you'll see science nerdy-like guys say something like, well, it's this twisty, laddery, ropey-like thing. That probably doesn't help you very much. It doesn't help you very much understand how this actually works, or even does this plastic model. The structure of DNA was first presented by Watson and Crick in April of 1953, commemorated in this issue of Nature. It was published in Nature now more than 50 years ago. And it was also commemorated in Life magazine with this article, or Time magazine, I mean, where the title was The Secret of Life. And this was actually Francis Crick's comment to what one of the people of the press asked him that they had discovered. And his answer was, the secret of life. That was what he could say about the structure of DNA. So why do we say that? Why did he say that? Why, after the discovery of the structure of DNA, does Francis Crick respond the fact that they had discovered the secret of life? Really? Well, let's take a look at that. DNA, the chemical name deoxyribonucleic acid. That probably doesn't help you very much either unless you're a chemist. Any more than this is a ropey, laddery, stringy-like thing. But I hope to convince you that the structure of DNA is ultimately at a foundation for understanding how life on this planet works. What does DNA look like? Well, if I were to take a sample out of you and put it into a Waring blender, let's say take your hand or something, and put it into a buffer and spin everything out, add some salt to that, and 2 volumes of cold ethanol, I could precipitate out your DNA. We'll all be doing this at the last lecture in this class. We can isolate our own DNA. But if I were to do that, DNA would precipitate out in terms of a flocculent, white, gooey kind of stuff, and it would look like this. Basically, it's fibrous. And Oswald Avery first did this and spun it out onto a glass rod. And if you ever want to do that, there is a certain wonder to this. You know, that you actually have this purified form of DNA, especially once you understand what it is. But this doesn't help you understand it any more either than this model, or saying it's a ropey, laddery thing, or its chemical name, deoxyribonucleic acid. So what was it about the structure of DNA that so compelled Francis Crick to say he had discovered the secret of life? Well, the well-known structure, the iconic structure of the double helix, is composed of this sugar phosphate backbone connected by these bases, these nucleotide bases that I mentioned in the first part tonight, designated A, T, G, and C, for adenine, thymine, guanine, and cytosine. So these are the chemical units that comprise information. Your computer, all your digital information is comprised of zeros and ones. And look at how much information we can store. It's amazing. George Church is a bioinformaticist, genomicist at Harvard. And he has just published a procedure on using the letters of DNA as a code to replace a binary code. So if you think you got a lot of information now in all this digital stuff, imagine A's, T's, G's, and C's, and we can look at all the information that is stored in a single cell and how small that is. So now you can put 18 gigs in your pocket. Imagine in the future computations done using DNA on nano or even pico structures. Things get pretty interesting. But life has operated on this code for quite some time. A's, T's, G's, and C's linked together in this sugar phosphate backbone. So here, the phosphate group is symbolized with this orange piece, and the sugar connecting them together is what the ribose is. Ribose is a sugar, deoxyribonucleic acid. So back to information theory. You need difference in symbols in order to store information. One symbol does not information make. So if we have a chain of molecules that is simply repeating itself, it really contains no information at all. Things get interesting, however, when those symbols change, even zeros and ones. So one of the hallmarks that I'll bring up in a subsequent lecture about life on this planet is that life assembles itself in chains. We've already seen chained molecules, like sugars make complex carbohydrates, amino acids make proteins, nucleic acids make RNA and DNA in these chained molecules. So difference itself becomes information. How those nucleic acids are arranged, that sequence contains information. A chain of links by itself, useful, but not that much information, right? Whereas if you look at the Morse code, is a chain of 2 units, dots and dashes, like digital code. So dots and dashes actually can contain a fair bit of information, zeros and ones, gigabytes, terabytes of information. Even our alphabet is pretty good at information, containing its 26 different units. We do reasonably well with information with those 26 units. Now is the winter of our disco. So the structure of DNA then lends itself to containing information, and a lot of it, in a very small space. So what was it about the structure of DNA that compelled Francis Crick to say it was the secret of life? I've mentioned that all the DNA in a cell is largely in its nucleus, but mitochondria and chloroplasts also contain DNA. But all the information for each protein is controlled by the DNA sequences. So by controlling the information for proteins, we control what a cell does. So, for example, you have eye-specific proteins that are regulated by eye-specific genes, meaning that they are on in an eye, but perhaps off in other cell types. You have liver-specific genes, making liver-specific proteins, and so on. You have genes that are on in every cell as housekeeping sort of functions. And the nucleus contains most of that DNA. The DNA in the nucleus is typically packaged into chromosomes. Humans have 46 chromosomes that come in pairs. You have 23 from your mother and 23 from your father. And each one of those contain genes. Some are dominantly expressed, some are recessive. So you have dominantly expressed genes from one parent, some are suppressed in another. For example, brown eyes is a trait that is dominant, and you can understand the genetics of that pretty easily, looking at your parents or perhaps your grandparents. Blue eyes being recessive. And there are about 24,567 genes in a human. I say about because that number has not been definitively nailed down, but that's pretty close. And genes are pieces of DNA on chromosomes. So you can think of your 23 chromosomes like volumes of an encyclopedia, where one chromosome might be analogous to a volume in a set of encyclopedias. In a genome, or all of the DNA in an organism, might be like the entire set. It's interesting to me that these comparisons are often made about the genetic code and our own language. I'll get back to that later, but I will also position that perhaps genes themselves behave very similar to information in our language or even ideas. And evolutionary pressure on genes is similar to the way it is on organisms, is similar to the way it is on species, is similar to the way it is on ideas. Why do some ideas survive and other ones go extinct? In the population of ideas, the pressure is the same on biological entities as it is on information. So, going with that analogy, a simple base pair in DNA might be like a letter. Okay, 1 of 26 in a letter, not much information. T, it's okay, we got a T, but it's not a big deal, right? You take 3 of them and put them together, T-H-E, the, pretty good. I don't know what it means all by itself, but you know, we got some information going. Same thing with bases. A-T-G, we got some information there. We've got 3 different symbols that now we're going somewhere. You string a whole bunch of these together and you get a sentence. Letters, I mean in our alphabet, you get a sentence comprised of words. Same thing with DNA. You string a whole bunch of A-T-G-C, A-C-C, so on. We have a language, a sentence that's like a gene. You string a whole bunch of these genes together and you make a paragraph. Now you got some more meaning, a few genes stuck together. All of them together, now you've got a chromosome and maybe a biological pathway. And so that's kind of how it goes. There's also a cut and paste function I'll get to later. But the information in DNA is transferred to an intermediate called RNA. DNA stays in the nucleus. It's transferred to an intermediate molecule, RNA, and then goes out into the cytoplasm where that information is transferred into proteins. And as I mentioned, it's proteins that do all the work in cells. Enzymes, for example, are usually proteins. So enzymes are proteins that catalyze reactions in cells. So again, what was it about this structure? We need to go back in time a little bit. 1950, 1951, we knew, humans knew, that DNA was the hereditary material. We knew about heredity from Mendel, but actually we knew about heredity before that. We have to give Native Americans here a little credit for discovering corn and understanding probably the early genetics that gave rise to that, and in Mesopotamia developing crops like wheat. But Mendel actually was the guy in the 1800s that deduced traits. And if you cross this plant with this plant, we can get traits communicated through heredity. Heredity then was linked to DNA in the 1940s. So now come 1950, everybody's thinking that DNA is the hereditary material, but no one knows what it looks like. And there were several different ideas about this at the time. Linus Pauling was a chemist who became later famous for his input into the chemistry of vitamin C and some other important discoveries, had put forth the idea that DNA was a single helix, like a helical stairway. There were other people who had hypothesized that it was actually a triple helix molecule, that there were 3 strands involved. And there were a couple of people who thought that it might be a double helix, but along comes James Watson. James Watson finished his PhD in biochemistry from, I think, Purdue University in West Lafayette, Indiana, and decided that the structure of DNA was the most important question of his day. So he took a postdoc in the UK, bounced off of one, and ended up in the lab of Francis Crick with the idea of purposefully trying to deduce the biochemical structure of DNA. So it wasn't like he just thought of this one day. He actually, and he was about 28 years old, I think. And there they are in this famous photograph in May. This is about 3 weeks after they published their article, 4 weeks after they published their article in Nature, in the Cavendish Laboratory in Oxford, with this iconic drawing in the background of the double helix and this model, which James Watson had put together as part of his postdoctoral research. And this is young Francis Crick, also his mentor and advisor. Anybody know what he's holding here? You don't see these around anymore. It's a clue. It's a slide rule. People used to use these to figure out complex mathematical equations that now we just do on a computer. But they published this article in the April 25th issue of Nature in 1953, and I pointed out their first paragraph to this one-page article. This is a one-page article. I would challenge any of you to write one page that could have this much impact. Aspire to that if you can. Anything, anything. A poem, anything that could have the impact of this page. And here in this paragraph, they say, we wish to suggest a structure for the salt of deoxyribonucleic acid. And they even abbreviated it D period, N period, A period. We don't do that anymore. This structure has novel features which are of considerable biological interest. I mean, no kidding, right? So what exactly did they mean, and is this just wry British humor? James Watson went on to write a book called The Double Helix, which describes in detail his account of the discovery of the double helix. It's interesting to read. It's a fast read. It's entertaining, actually, if you're not a scientist, or actually even if you have an interest in science, how this was actually done. I mentioned creativity and a fast pace. But you know what? I fault this book deeply for leaving out a very important and central character in the story. At the same time that they were working on this, Watson visited the laboratory of an X-ray crystallographer named Maurice Wilkins, who he invited to his lab to give a description of his work on X-ray crystallography. In parallel, there was a woman who was also an X-ray crystallographer working at the same time. Her name was Rosalind Franklin. And Rosalind Franklin was an X-ray crystallographer also taking pictures of crystallized DNA. Now, I'll tell you something. You don't have to be an X-ray crystallographer in order to understand the data here. And Watson was an astute guy, and he visited the lab of Rosalind Franklin, who was taking pictures of crystallized DNA that she had made from calf thymus, and he saw this image. And I mentioned that one of the hypotheses had been a single helix, an alpha helix, posed by Linus Pauling. In an X-ray crystallography image taken with an electron microscope, you bombard that crystal and look at the diffraction pattern of electrons that come out the other side, of X-rays that come out the other side. And if it were a single helix, you see these bands that are coming out here? If it were a single helix, you would expect these to diffuse out around like this. If it was a triple helix, you would expect another set of bands right here. So this image explicitly shows that DNA is a double helix. He visits her lab, runs back and puts together that famous Tinker Toy model, not Tinker Toys, but you know what I mean, that we see in that journal article. So James Watson, Francis Crick, and Maurice Wilkins go on to win the Nobel Prize in 1962. Where's Rosalind Franklin in this story? She was also conveniently airbrushed out of James Watson's account in the double helix. Why? Well, one, she was a woman in the 1950s as a scientist. Secondly, she decided to publish her own work independent of them. And we see this in exactly the same issue, just a few pages away from their article. Nonetheless, Watson doesn't even refer to her in his book. Really? So I think most people know now the contribution of Rosalind Franklin. And by the way, Nobel Prizes aren't awarded posthumously, and she died prior to 1962. Now DNA has become an icon. Actually, we think now that it pretty much represents science. If before a microscope might have represented medicine, DNA is all over the place. The UK even minted a coin with the double helix on it to remind us that it came from a British lab, I'm sure. But here it is in a painting by Salvador Dali, titled "The Butterfly Landscape, Life Coming Out of the Double Helix in the Desert." This is 1958. Salvador Dali describing DNA as life in the desert even before the Nobel Committee recognizes Watson and Crick. And we see it like in this sculpture in the atrium of a hotel, or here in this sculpture that's out at Cold Spring Harbor. So who were the major contributors to the discovery of DNA? Watson, Crick, Wilkins, and Franklin. The secret of life? Why? Well, I think in order to understand this, not only the double helix structure itself, but we get a clue in a paragraph that occurs towards the end of that one-page article. And it's right here. It has not escaped our notice. So it's kind of like at the end of the article, by the way, it's not escaped our notice that the specific pairing that we have postulated immediately suggests a possible copying mechanism for the genetic material. All right, you might think that's subtle. Your ones and zeros and terabytes of information on your computer are one thing, but it doesn't copy itself so well. What they postulate in this double helix structure, not as just the structure of DNA, but it describes how DNA can be copied. Every single time one of your cells divides, it's got to copy all of your DNA so that each cell gets an exact copy. There's an exact copy of your DNA in every single cell in your body. That is phenomenal. How does this happen? And that is a very cool statement. By the way, it's not escaped our notice. So when they say that A's pair with T's, the specific pairing, on either side of the double helix, A will pair with T no matter what side it's on. G will pair with C. Therefore, the copying mechanism is that if the middle of this double helix unzips, A will pair with T's, and this will form a template out of which you can make two identical molecules. This thing comes apart, the other one fills in A's, T's, G's, and C's, and you have two identical molecules. Brilliant. Over billions of base pairs later. So before a cell divides, all that information that represents the information to code for a whole U is copied. And that's occurred several times since we've been talking here today. So the DNA is copied as the double strand sort of unzips, like a zipper, exposing a template on each side so that A's will match with T's and G's will match with C's, and therefore this template and that template are copied exactly. But wait a minute, that sounds so easy. DNA just does not copy itself on its own. If I isolated that DNA out of you and I sat it on the shelf, it would sit there for the next few thousand years or so and not do anything at all. How does DNA actually copy? DNA is copied by using different proteins. Now this is interesting. DNA itself codes for proteins that copy itself. We've got a chicken and the egg problem right there. But in any case, we'll get back to that later. There are a whole series of proteins that are involved in copying DNA, represented here by these cartoon characters. We've got one that initiates where DNA will start to copy. It just doesn't start at the end and work its way from one end to the other. There are actually initiation sites. This protein recognizes one of those initiation sites and the DNA copying process starts. Another one called a helicase unzips the DNA, bringing apart those two templates. Another one that's very important for biotechnology, and I'll get back to this later, is called a polymerase. It brings in the A and matches it with a T. It brings in a G and matches it with a C and builds the strand, polymerizing that strand together. There's another one that's a repair enzyme. Bring in an A that matches a C, whoa-oh, we got a problem. And this one actually will recognize that mistake and fix it. So that's a repair nuclease. There's a topoisomerase, so-called, that's an unzipper. So if you just take that helicase and you take DNA and just open it up like this, it's going to spin around at the other end into a big goo ball, basically. So this enzyme sits at the other end of the molecule and turns it in the other direction to make sure that that doesn't happen. You've got these single-stranded DNA binding proteins that at some point that DNA is going to be in a single strand and very fragile. If it breaks, we've also got a problem. So there are these proteins that will bind right to that single strand as this thing is polymerizing and working its way through. And then there is this one called a ligase that stitches two broken ends together. This is also a very important enzyme that I'll talk about later. In fact, I want to bring up a point right now about biotechnology. Most of what we're going to be talking about in biotechnology was not invented de novo by humans, but rather as used components of life in order to work with life, particularly this one and this one. So this then together works as a high-speed, high-fidelity information copying system, and hopefully it makes very few mistakes. But mistakes do happen. They do. Sometimes an A goes in where a T should go. And this creates problems on the one hand. Certainly this is the basis of a lot of genetic diseases. Certainly diseases like cancer are involved in these types of mutations. These happen all the time. They happen due to environmental insults. They happen just due to random copying errors. These happen when you're making eggs and sperm and are transmitted than inherited. So certain hereditary diseases are mutations that occurred a long time ago in a family and are passed down a few generations. But mistakes do happen. And there's another thing about mistakes that actually contributes to this: variation. All of you are slightly different. Yes, it took a PhD in biology to recognize that fact. But you're all different because you vary just slightly in your DNA. Not even by a lot, actually. You're 99.9% the same at your DNA level. But that 0.1% makes the difference that I observe, and a few that I can't. There are enzymes that are involved in DNA repair. And if you get mutations in those genes that code for DNA repair, now you've got another problem. Oftentimes those mutations are involved in cancer too. So say, for example, we know that UV light causes skin cancer and melanoma. We know that recently. UVB light can cause two T's to stick together, thymidine dimers. They don't copy so well, and those can make mistakes. So there are enzymes that can come in and fix that, repair enzymes. If you get a mutation in a gene that is involved in repair, now you've got an even bigger problem. Okay, so information then can copy. That's a fundamental fact of life. Information is an information processing system capable of variation. So, how do we then decode the information in DNA? Well, if DNA codes for proteins, we go through this intermediate called RNA, which is ribonucleic acid. So instead of deoxyribonucleic acid, it has a slightly different sugar molecule, ribonucleic acid. And what happens here to decode a gene is the DNA is again unzipped, exposing a single-stranded template on either side, and an RNA molecule is then synthesized off of that template. So the template, A's, T's, G's, and C's, is transferred to a complementary strand for that information comprised of RNA, which then leaves the nucleus. And instead of those several cast of characters that I just showed you for copying DNA, RNA polymerase is a single enzyme that opens up the strand, brings in the different bases, and makes the RNA strand and then closes it back together. So RNA polymerase makes the RNA out of the strand of DNA that we call a gene. That RNA then leaves the nucleus to code for a protein. The only difference between RNA and DNA, besides its single-strandedness, is that a U substitutes for a T. So this is a molecule called uracil. Once I was talking about this to a bunch of 8th graders, and an 8th grader actually asked me, Dr. Kausch, why is uracil used instead of thymidine? So maybe you should be a grad student. But actually, this is a good question. You know, you think about it, most RNA uses uracil instead of thymidine. It's structurally more stable in a single strand and is selected for that reason. So RNA has a different sugar backbone and uses uracil instead of thymidine. You might want to practice taking a DNA sequence and looking at what its homologous RNA sequence might look like this, for example, where A then would pair with a U, G would pair with a C, G would pair with another C, C would pair with a G, T, A, G, G, U. So DNA stays in the nucleus and RNA leaves. Realize that this means that RNA can be temporal. You want RNA to come on when a gene is needed to be made into its protein and then off, but the template stays in every cell. It's a good way to control information, right? We call that process of information transfer from DNA into RNA transcription. And this is a pretty, I think, interesting diagram of an illustration about how that might occur. DNA stays in the nucleus, RNA leaves the nucleus to be decoded into a protein in the cytoplasm. The RNA message leaves the nucleus and is decoded in the cytoplasm using this cellular structure called a ribosome. A ribosome takes that messenger RNA and is decoding that sequence using triplets of information. So I mentioned the word the, T-H-E, doesn't have much information. Similarly, ATG will code for one amino acid in a protein. If you string a bunch of these together, you can spell for a protein. I mentioned ATG. ATG spells for the amino acid methionine. That's 1 of 20. Life on this planet uses 20 amino acids. In fact, there's more than 20 amino acids in the world. Why 20? But the code for those amino acids is the same for all life. So in other words, ATG codes for methionine in you. It codes for methionine in a pea plant. It codes for methionine in a cow or a whale or anything else. So if I take the genetic code for any protein and move it into another organism, the code stays the same. The language is the same. This is very interesting. That means I can take a gene out of you and put it into a corn plant and it'll still make the same protein because the language is the same. Got it? Translation also requires many different components. Translating the language of RNA into the language of protein requires the mRNA, the message, these transfer RNAs. These are also encoded for in the nucleus. There are 20 transfer RNAs that link up the amino acid and bring it into place to match the template. So these have the corresponding code that brings in each one of the 20 amino acids. There's a ribonucleic acid that is in the ribosome, or rDNA, that makes up the ribosome complex, the 20 amino acids, and these molecules for providing the chemical energy involved in making the protein. So these transfer RNAs have these clover-like shaped structures, and these are also comprised of amino acids. And the code is in triplets. So the template in DNA is transferred to this mRNA, and then 3 of these are spelling for 1 amino acid, and so on. So the code is read from one direction to the next. So if this protein is going to be, say, a muscle protein, that has a different code than, say, a metabolic protein. And the code that's spelled for here is directly transferred to here and is decoded, and it's those different amino acid sequences that make each protein different, each gene different. So your gene for your hemoglobin has a different DNA sequence than the gene for some other protein in your system. This code was first deduced by some people working in Bethesda at the Cancer Institute, originally making a synthetic code. And I want to point out something about this particular chart, is if you have the first position on this side, the second position on this side, and the third position here, here's that one A, U, G, or in the DNA it would be A, T, G, reading for methionine. So AUG spells for methionine, and that's typically the start site of any gene. AUG will code for methionine. I will also point out that the code is redundant. CUG codes for this amino acid, and CUA will code also for that amino acid, leucine. So the code is redundant. Thirdly, there are stop signals. UAG will code for a stop, so will UGA code for a stop. So you have a start, a redundant code, and a stop for 20 different amino acids. And that code is the same in plants, animals, and fungi and bacteria. AUG codes for methionine in all those different organisms. And therefore, we can make proteins that way out of genes. So what do we mean when we say that a code in DNA is a gene for a protein? We used to think that one gene codes for a protein. Your 24,457 or so different genes, there are over 100,000 proteins. That means that one gene can code for more than one protein. Things get a little more complex. But mistakes do occur. So what happens when a mistake happens? If we have the original DNA code in this template here, coding for this amino acid sequence, here we have our AUG coding for methionine, UGU coding for cysteine, and then UCC coding for this amino acid serine, we get one particular type of protein that will have one particular type of function. What happens if we made a mistake and we have a mutation in the copying function? As shown here, let's say we leave one out. We have AUG still codes for methionine, that's normal, but we leave out this G. We're still going to read things in terms of 3. So instead of reading the UGU and getting cysteine, we read that UUU, which now codes for this amino acid, phenylalanine, which is different. What does that mean? That means that that protein is not going to be the same as that protein. That's either okay or it's not. Most of the time, it's not. And that's the basis for a lot of genetic disease. Or sometimes it's just benign and it doesn't matter. Sometimes, given a redundant code, it could actually be the same. We would call those silent mutations. So that's what you have. We call that a point mutation. And those can either be because of a deletion, or you can have an insertion, which would also cause a frame shift. So here you have your AUG, and then if we have an inserted one here, everything gets shifted down and we have a different amino acid sequence once again. So insertions, deletions, frame shifts, this is how we move information around. So life is an information processing system capable of replication with variation. I'll start that as my definition of life. And mutations are the basis of that variation. And all of you are slightly different because there is variation in your DNA. Some of it large, some of it small. And the sequence of DNA is what is a gene that specifies the sequence of amino acids that spells for a protein, and it's proteins that do all the work. And the chain of amino acids that is spelled for in a gene derives its function out of its shape. The shape of a protein like hemoglobin is meant to carry oxygen around, whereas the shape of a protein that makes a muscle protein is a different shape and has a different function. And that shape is controlled by the sequence of amino acids. You change that one amino acid and the shape changes ever so slightly, and that protein either does its job a little bit better or a little bit worse, or perhaps even not at all. What happens, for example, if one of those amino acids, instead of changing to another amino acid, changes to a stop? Right in the middle, you've got a very short protein, and that probably is not going to do its job. Also, so you have 24,000 genes or so. They can't all be on at the same time, or else you're just a bag of protein, right? Just a balloon full of protein. It's a big deal. You have to have a way to turn certain genes on and off. Some are on during early development, when you're an embryo, and off later as you're an adult. Some are on to make eye cells and have eye function. Some are on in liver cells and off in eye. So there are switches that we call promoters that regulate when genes are turned on and off. Typically, these are upstream of a gene. Just 2 weeks ago, a large group of scientists, about 500 or so, published about over 20 articles describing what was previously called junk DNA. I mentioned that you have about only 2 to 4% of your DNA as genes. What's the rest of it doing? For about 30 or 40 years now, that's been called junk. You know, nature doesn't make junk very often. It doesn't persist. We learned just a couple of weeks ago that a lot of what was called junk previously is this regulatory DNA. We've known about promoters for a very long time. Segments of DNA that turn genes on and off. When I say a gene is on, I mean it's making its RNA and it's making its protein. This is a diagram of a simple bacterial cell, a prokaryotic cell, maybe 960 so genes or so. And each one of these different colors represents a different protein. So you can see that each one of these proteins is encoded for by a gene, some of them moving things into and out of cells, some of them involved in metabolic functions. Yeah, memorize this for the next quiz. I'm just kidding. But what I mean to say is that one function of life on this planet is this flow of information. It's consistent. Information by itself isn't enough. this flow from DNA to RNA to protein is pretty much consistent. You will meet some viruses that actually are RNA, influenza, for example, HIV. They get into cells and engineer a cell to actually convert it into DNA. So they run backwards for a little brief time. But basically, the flow of information on this planet is from DNA to RNA to protein. I'll just leave you this to read, but this is my conception of life as a "This I Believe" essay. I think you're instructed to do these in Writing 101 or 104. Well, I believe in life, so you can check this out on the website. Whoa. Right? Hey, are you melted? Are you melted yet? That was something. But look at it this way, right? That was a lot of information. But now you know how life works, right? There you have it. We're not done yet, I'm afraid. I know this is a long one tonight. Bear with me. We can take a brief break, though, and we'll come back for, I just have to cover some techniques on biotechnology before our quiz. So another brief break. Thanks.